So you have the presentation, and Dr. Doorn, again, I'll just go through it quickly. Michigan native from Grand Rapids, Calvin College for undergrad, University of Michigan toxicology for graduate school. Postdoc looked at, in the University of Colorado, looked at alcohol-induced liver disease. First faculty position at the University of Iowa, 2004. Has had his whole academic career there then, and is currently tenured professor in the Institute, in the Pharmaceutical Sciences and Experimental Therapeutics Department. A leader in both the P30 Center that they have there, NIEHS P30 Center, and the P42 Superfund Center, working with that too. Today, he's going to talk about mechanisms of neurotoxicity of organochlorines, and particularly the role of astrocytes. Welcome, Dr. Doorn.
All right. Yeah, thank you for that very kind introduction. And can those on Zoom hear me okay? Wondering how far I can step back from the microphone.
Yeah, we can hear you.
We can hear you.
Okay, fantastic. Okay. Well, yeah, thank you for that wonderful introduction. And yeah, just a couple of points. I've been at University of Colorado, University of Iowa for about 19 years, almost 20 years now, and it's a wonderful university, great place to be, College of Pharmacy, I have an administrative appointment too as a DEO, so I've enjoyed this time being away, the visit, talking science, and I'm sure as I haven't looked at email in a while, I probably got like about 100 messages, problems. Actually been getting some text just before this seminar, and you saw me put my phone back there, said enough. So it's been wonderful to be here, hear about all the exciting projects going on, and your center is fantastic in terms of the instrument capabilities, the collaborations, the P42, P30. Yeah, it's remarkable. So I've learned a lot. It's been a fun visit. And also just a note too, yeah, so I'll be talking about today, I've basically, two main projects, and one of them, you heard some of my background. I actually spent some time University of Colorado as in an alcohol research center, and it was during that time I learned about aldehydes and metabolism, and then that kind of then it spurred some of my later research looking at reactive metabolites and how these might be, maybe, components of a disease process, or, perhaps biomarkers for earlier disease diagnosis. So, all right, so let's see how to continue on. There we go. Okay. All right. So, so today I've got two themes here I'll talk to you about, and my title is I can go back here. Okay, so the title what you saw is astrocytes, neurotoxicity, but I'm going to be adding some themes to that. Oh, here we go. And thinking about really about thinking about is mechanisms of toxicity. And for many years been very interested in the environmental basis of neurodegenerative disease, the environmental basis of disease. How does maybe it's gene environment, but how can the environment, especially if you think about the sort of myriad of pesticides, environmental pollutants, and and there's some kind of interesting observations that the correlation between exposure and later disease. And if you try to think about chemically, I have a little bit of me at heart as a chemist, and so I think, well, is there anything that structurally can put these together? Is there anything structurally related we can figure out a target? And in some cases, not really. There's very structurally dissimilar agents that may be risk factors. Don't use the word cause, but risk factors for similar to conditions, diseases, neurodegenerative disease. And the question is, how can it be? Is there a common target that might be at play here? And so yeah, so two themes. First one here is cell selective toxicity, metabolites, organochlorines. And this is and actually what I'll be talking about today is work over the last 6 to 12 months or so. I've gotten involved with our last few years, our Superfund research program at Iowa, and the focus of that is polychlorinated biphenyls. And in particular, airborne PC, I was calling PCBs. And I was brought into this as, you know, they're going for renewal, a new project, and interested in neurotoxicity or neurodevelopmental outcomes. And so I joined the effort, and this P42 have been running for many years, you know, as far back as I can remember. But I joined, and you know, and I was learning the PCBs. I'm still learning the PCBs. You know, in terms of their history and toxicity, this is relatively new to me. But I'm really excited about, you know, some of our recent findings and in astrocytes and, you know, particular target in the mitochondria. And yeah, you know, you'll know what gets me excited is, again, thinking about, you know, is there cell selective toxicity? Are there targets? What does this have to do with the structure of the molecule? The metabolites, and that's why I'm gonna weave in a little bit of some of our other research that we're involved with. That's funded by NIHS, was thinking about you know, biomarkers and aldehydes and production of aberrant levels of aldehydes, particularly metabolites of neurotransmitters, as being you know, somehow a piece of the puzzle for pathogenesis. But I'll talk about today is their utility potentially as a biomarker. And so much of this is some of this work is published. Published work I'm just going to say a few words about, but much of this is actually unpublished, and I feel like a perpetual student because I'm still learning. So let's see if I can advance this here. All right. Not sure, do I hit the up? Oh, here, the wrong way. Let's try this. Here we go. Okay, so some gaps in the knowledge here. If I can read my own writing here. You know, interested in these organochlorines, PCBs, and as I'll show you, one thing that's I think is unique is that we're in Iowa in terms of the PCBs is we have this fantastic synthetic core. And so, you know, so we can ask questions, well, what about mixtures? You know, but also we can ask questions about individual congeners and metabolites too. And so we've been able to, synthesis core has been able to make some of the primary metabolites, and ask the question, are these part of the mechanism too? Are these, you know, the ultimate toxicants? And then the last the second theme, talk about catecholamine metabolism, environmental agents interfering with dopamine metabolism, in particular, some metabolism information of aldehyde. So asking that question about a biomarker. All right, see if I can go down here. I'm not sure, do I push the right button or the up button there? Pushing both here. I wonder if there's an easier way to advance these slides. No, the keyboard kind of works, but if I hit the down button, it doesn't do anything. Hit the right button. Hit the up button here. Here, you know, actually, I think I can. Oh, here we go. There we go, in the corner. There's a. How about that? There we go. Okay, I'll just won't move here. So in terms of, you know, goals here, elucidating mechanisms of toxicity, novel targets for therapeutic intervention, biomarkers are, you know, really some of the themes here. So, all right, so I'm gonna start off with this part one here, and these PCBs. And there's been a lot of really interesting literature reviews, you know, many of them written by people in the Superfund Center, which I'm part of, which I know you have one here. And for those of you involved, you know, it's really a delight because, you know, we've got our basic science, you know, projects, but also there's a environmental exposure component too, and bioremediation. So there's all these facets to it. So it's been a wonderful learning experience, but also collaboration as well. So yeah, so there's been a lot of epidemiological evidence saying that the, you know, PCBs are bad, you know, carcinogens, neurotoxicity. There's this great review from Bullard and others, systematic review a couple of years ago, I guess of last year. In terms of some of the mechanisms, you know, maybe thinking about things such as ryanodine receptor, dopamine homeostasis is which caught my attention, which brought me onto this project, and something we're still trying to close the loop on. but also too, in terms of the, what's been really fun is thinking about metabolites, metabolism. That's been one of my real interests. And, if you look at this particular PCB here, 52, what you'll see is that there's a hydroxy, there's a sulfate metabolite, some of the, the major metabolites that are, that are formed in humans, especially in this 4-hydroxy. So that's the question is, what impact do these have? And in terms of the mechanisms of toxicity, if you look in the literature, neurotoxicity, PCBs, you'll see a lot of, the majority of the work deals with, neurons, in vitro, in vivo work, looking at neurons, neuron cells, cell lines, and culture, but very little work exploring this idea about the glial cells and astrocytes, microglia. And it's just fortuitous. It was actually just before COVID hit, I was at a Society for Neuroscience meeting and kind of wandering along aimlessly, among the 20,000 participants, and happened upon this poster of this Mandana, and she showed some interesting data on PCBs and astrocytes that, there's some toxicity, there's ROS formed, and seems to be some metabolic changes and changes in bioenergetics. But she was using mixtures, and I thought, well, it's really interesting work. And so this is kind of what spurred some later work where rotation projects. Hey, let's look at astrocytes and PCBs. And there's been plenty of work with neurons, and that's where we made some really interesting findings that I want to tell you about today. And so asking these questions, of course, the astrocytes being very important cells that, I had to convince myself that, astrocytes are something really important and we need to work on them. And yes, they are really a fascinating cell type, thought to be just sort of this, this glue or this dumping ground. And in some regards, they do act a little bit like a dumping ground from neurons, but they have a fascinating role in terms of, ions, metabolism, neurotransmitters, blood-brain barrier, and they talk to other cell types too. And so it's a very important cell type. And let me just next slide here. Okay. So so in terms of the PCBs, we asked this question, well, are these following up on this work, are these PCBs, are they targeting astrocytes? Is there any interaction? And the first question, toxicity, not a real interesting one. In my opinion, it's not so much interesting that the cells, die. It's that, okay, the cells that, are alive, what's happening in them. But you get some values like IC50s, and it tells you a little bit about, okay, well, how are the cells responding? And so what we're able to do is look at some of these, the center is very interested in airborne PCBs. And we have this Chicago air mixture and school air mixture. And I found some of the major congeners that are involved in this, one of them being PCB52. And we see a few of these other ones as well. I'm gonna wander out a little bit here. So we see things like PCB 311, 25, 28. So we asked the question of, okay, well, which PCBs? We've done some work with mixtures, but individual congeners. What we found was really interesting was that this PCB52, which is thought to be a major component of these air mixtures, seem to be having an effect on these astrocytes. The astrocytes were responding, and what was really interesting was that the hydroxy metabolites are actually more potent than the parent compound. And also too, what was interesting was that if you look at the sulfate metabolite as well, that was for the 52, that was the one that seemed to be the most potent. The cells were responding to this PCB 52 sulfate. So we got some idea about structure activity relationships here. It seemed like the cells are most sensitive to this PCB 52, and then to these metabolites. And we can see some of these IC50 values here. And what's interesting, it seemed like overall the hydroxy was the most potent for all of them, but it's unique for this 52 was this sulfate metabolite here. And so we'd like to make some judgments about, okay, well, maybe more chlorines, maybe structure, and we haven't thought about anything about the spatial conformation, but But again, what we see here is it seems to be something unique about this 52, which was something that the center is very interested in looking at. And so just a couple other things too is that looked at this MTT cell viability and then cytotoxicity, and we saw this this correspondence here between the two, and corresponding toxicity, they seem to correspond at some point. But one thing that was also really interesting too is part of this PCB, part of the Superfund project in the center is that we have this group involved in environmental exposure analysis, and engineers looking at bioremediation, looking at ways to measure, but also they're very interested in modeling. Okay, how do these agents partition, say, in soil, water, air, but also too in terms of cells. If you have you add culture, where are these modeling, where are these agents going to go? And so just a couple of things. I'll work from the bottom up here, but we're able to model. Okay, well, for all these agents, and this is the work of Andre Martinez. So I can't take credit for this, but they looked at lipids versus liquid versus protein. If it's inside the cell, where is it going to go? But then also too, if a cell sees some of these agents here, what's going to happen? Is the cell going to take them up or not, or where are they going to go? And for the most part, there's really, these predictions, are probably not all that exciting, but one thing that was kind of interesting was that this PCB sulfate, if you look at this prediction here, it really shouldn't be taken up by the cells. It shouldn't be in the cells here, but we're seeing some, measurable toxicity, some measurable response from it. Slide here. All right, here we go. And so these data we've been using the C6 astroglial cell line as our model simply because they're easily acquired, and we've had it seems to be a fairly robust cell line, and literature there's good evidence for it. But we did want to look at primary cells as well, and that's the question: are we seeing similar toxicity, or is the cell line uniquely sensitive? And so we obtained primary mouse astrocytes as well as primary rat astrocytes. The mouse astrocytes are actually from Ron Tulkins at Colorado State University, provided us with those. And then my student and a postdoc, Mia and Laura, isolated the rat astrocytes, and so we looked at toxicity here. And for the most part, it looks pretty similar that, these primary cell lines are responding just like the cell, these primary cells are responding just like the cell line. But one thing that was rather unusual about this was these are problematic dose response curves. And if you go back to the earlier dose response curve, the C6 culture here, you see, very, very pretty dose response curves that you'd see in a pharmacology textbook. But as we move to these primary cells, we see this, seems like there's almost this increase in viability, this precipitous drop off, and my student Mia could not find the intersecting point to get to be able to calculate an IC50. And one thing that we observed was that, this is so these cells are 80% pure. So if we do, as noted above here, if we do flow cytometry, GFAP, IBA1, these are about 80% cultures, so there's some contaminating microglia in here. But one thing that was interesting was that, we asked another question too in terms of purity, but also we wondered about, is there any sex-dependent effects here? And so Nia and Laura were able to isolate rat primary astrocytes and have them typed via sex, and these are pure cultures, about 95% pure astrocyte, and so the answer is no. We didn't see any differences, and these are from PND 1-2 rats. Perhaps if we let them mature longer, or if we added some estrogen, perhaps to push the sex. maybe you'd see a difference, but we didn't. But one thing we saw was that when we purified, when these cultures are purified to say 95% homogeneity, we saw these beautiful dose response curves showing up. And so this raises the question, with these last dose response curves, what is going on? And I'm going to leave that for now. But the question is, well, does this mean anything about microglia or other cell types? I'm going to leave that thought with you and just continue on here, some of our other work. And so we asked the question of, okay, well, what is the mechanism here? How are these cells responding? What's happening, in these cells? And the first thing that we saw is it's a little bit hard to see here is that, just after a couple hours, low micromolar exposures here, I think these are the LC10 values. I wish Neil were here to my student to ask that up, because I can't remember if it's LC1 or 10, but we saw, it's just after 2 hours, if you use like mitosoc cell rock staining, you start seeing ROS you see ROS produced, and we got our positive controls antimycin, and then down here looks like metadione. But yeah, we see the ROS, and it seems like it's more of the the mitochondrial ROS than the total cellular ROS. So it seems like there may be more mitochondrial ROS that is forming. And so my student, quantified the ROS, just total ROS in the cell, just a crude way, DCFDA, and showed that sure enough, quantitative. So we can see qualitatively there's ROS forming, but quantitatively too, we're seeing ROS that is forming. And so we asked the question, well, is the ROS important for the mechanism of toxicity? Maybe it's the PCBs are just causing ROS mitochondria, and that's what's killing the cells. And so the answer to the question, well, based on these data, I can't quite answer the question or prove it, but what we thought was, okay, well, maybe what if we load these cells up with like a mitochondrial antioxidant? So we use MitoQ and MitoTempo, which have a TPP tag on them, and it routes these antioxidants to the mitochondria. You have to be careful with those because they're charged and they can cause some changes in gradients as well. But we're careful. We talk to the oxidative stress people on campus, free radical radiation biology people. And to answer the question is, these mitochondrial targeted antioxidants had absolutely no effect on viability. It didn't seem like they were protecting. And so it really raises the questions, well, what's going on with the mitochondria? And perhaps the ROS here, maybe it's just sort of incidental, maybe this is secondary to what's going on. And so we thought, okay, well, yeah, it seems like there's mitochondrial ROS that's forming. So cells are sensitive. We're seeing mitochondrial ROS. And there's some differential, potency here. It seems like hydroxy is more potent than the others, hydroxy metabolite. But we wondered about, okay, well, what about the cellular bioenergetics? Is there something that's mitochondrial going on? Well, the best way to answer that is to think about do mitochondrial stress test and think about, okay, well, are we changing the mitochondrial bioenergetics? And so we employed use of our I have a really great metabolic core that has a seahorse XF analyzer. And so we treated my student Nia treat the cells for went from a half an hour to 2 hours, and then we looked at how are the cells responding. And what we saw was that if you compare to the the vehicle control, we saw that these cells were responding. There's definitely some changes in the mitochondria bioenergetics. And this I think this was a 2-hour pre-treatment, but you see even with this, like the basal respiration seems to be lower here for the PCB for the parent compound. And my student went back as far as she could. I think half an hour is all that I think as fast as she could go, treat the cells and then start the analysis. And she would see the same thing. It's whatever's happening appears to be fairly quick. But we see this change here in terms of this basal rate. See looks like a proton leak. Seems some changes in terms of spare respiratory capacity. And I always get asked about why is this a negative value? The negative value for how they calculate spare capacity. And it just so happens to be, it drives me too, because I don't know what a negative value for that means, but it's just how they calculate spare capacity between the changes in the two phases. It just works out that way. So it seems like it appears that there's some changes in terms of bioenergetics. You use like JC10 for looking at mitochondrial membrane potential. FCCP is a positive control here. You also see that, especially with the hydroxy metabolite here. You see changes in terms of the mitochondrial membrane potential. Over the course of 2 hours, we're losing mitochondrial potential. And so it appears that the PCB, the 52, hydroxy, the sulfate here, conjugate metabolites here are, again, they're affecting the mitochondria, affecting the astrocytes. It appears like the mitochondria may be a target. We see changes in the bioenergetics, and so we thought, okay, well, what is really, what is the mechanistic target, and what are the full ramifications of this? And so we collaborated with a University of Iowa, a professor in neuroscience pharmacology named Stefan Strack, who's really an expert in mitochondria. I'm very interested in terms of mitochondria, individually, but also in terms of the networks of mitochondria. And as I've been learning about astrocytes, mitochondria, what's interesting is these mitochondria actually, they're not always in isolation. They form networks together. There's branching, there's interactions, there's networks, and these networks are, you call them a reticulum, and they're supposed to make more efficient your sharing metabolites, sharing of ions, and these networks are very important. And so the question is, okay, well, how are these? What's the effect of these PCBs on the mitochondria? What are some of the targets? And so anyway, so back to the story here. We collaborated with the Stefan Strack and neuroscience and pharmacology to look at more in depth what's going on in the mitochondria and changes. And he has this wonderful assay. And so you stain the the mitochondria with HSP60, and then you stain with nuke blue here, but stain with HSP60, which is a resident mitochondrial matrix protein. And from that, you can get kind of an idea of, what's going on with the mitochondria, where are the mitochondria, how are they interacting with each other? And you generate these really pretty pictures. And to someone that's not an expert in, mitochondria, I look at that and say, well, that's a really pretty picture. But I say, but some of the, my student who's learned much more about it than I did, and then, I'd ask her, well, these look different. What does that mean? And she's like, well, let's find out. And so what she's able to do with Stefan Strack is, he's got some imaging software, high magnification, and able to ask questions, look at individual mitochondria, and be able to do measurements, looking at length to width. They call it the aspect ratio. Looking at the mean mitochondria per cell. They count a certain number of cells, mitochondria. Also able to look at, just the length, the number, the area total, they call it the area squared. There's also something known as the form factor, which looks at, perimeter, as well as area, and takes into consideration branching. And so I'll just go through each of these here. It looks like that TV, oh, there we go. All right. So we see here with these, and I just have it highlighted in red for kind of an executive view. What we saw was in terms of the mean number of mitochondria per cell, it seemed like it's trending like these PCBs are causing an increase in the mitochondria, but nothing was significant. In terms of the mean mitochondrial length, it seemed like there was a decrease in terms of the following exposures in the 2-hour exposure. The area seemed to change as well. The area weighted, the form factor and the form factor here, and these are measures here of the branching and the complexity of the interaction, and you can see that in these pictures. And an untrained viewer like me, you can say, oh wow, they just look pretty, they look different. But what you see is that for some of the PCBs, when you treat the cells with a few of these PCBs, you're seeing a real change in terms of how these mitochondria are interacting with each other, changing some of the networks. And then this aspect ratio as well. changes, which is the length to width here. So it seems that there's some changes in terms of morphology. The last one here is a quality control measure, the form factor, the aspect ratio. It makes a straight line, and that's actually a good control for them. And to be quite transparent too, this is I'm learning as well too. The form. If you press me, what exactly does that mean? I would say, well, let's look in the literature, but it is a quality control measure. So one last thing I do want to mention in terms of the mitochondria. I thought, well, it seems like that is a target, but is there anything else that we can do? And I learned this trick from his name is Rodrigo Franco. He's a professor at University of Nebraska, Lincoln. But he caught me a while back when trying to find all these mitochondrial experts. Say, hey, what can we do? What can we try? And he had this great idea. He said, there's this trick that you can do with mitochondria. And so these astrocytes are highly glycolytic cells. They do utilize oxidative phosphorylation, but they're mainly glycolytic. And so advice was that if the mitochondria are really important for this mechanism of toxicity, if you force these cells to rely on the mitochondria and you see a shift in toxicity, that might give you some more information. So if you shift these cells to be more mitochondrial dependent and you see no change in toxicity, that probably means the mitochondria aren't that important. But if you do see a shift in terms of toxicity, that could indicate that this is some kind of a mitochondrial dependent mechanism. And so the trick here is that you culture the cells in glutamine. What eventually that does is that forces these astrocytes, are highly glycolytic, to start shifting more of their energy production to oxidative phosphorylation rather than glycolysis. And sure enough, if you do that, galactose, and also use the sugar galactose. So you remove the glucose, add the galactose instead, and culture them in the galactose, and you push them towards oxidative phosphorylation. Well, sure enough, this galactose, pushing into oxidative phosphorylation, we see a change in terms of the toxicity. It seems to be in terms of the toxicity curves, it'd be a leftward shift to lower IC50 values, which, again, seems to indicate that the mitochondria may be important for this mechanism of toxicity. All right, let's move on here. Okay. Oh, I forgot to mention too that one thing that seems to be happening for these PCBs, this is PCB 52 hydroxysulfate, is it appears that this response is rather quick. What is happening within, a lot of our endpoints are at 2 hours. We've done down to half an hour for the mitochondrial stress test, but 2 hours, definitely at 4 hours. Within 24 hours, we see apoptosis. And actually what's interesting too is we measured caspase 3,7 activity. And it seems that with this 52, the parent compound, the hydroxy, that you see this, this rapid increase in activity of the 3,7 here, even compared to, we use the positive control staurosporine, which, you do see that response, but 1 hour we see a response, but even by 3 hours. So whatever's going on with these PCBs here is happening quite quickly. So all right. So from this point, presentation here again, these are very very new data. Some of this is published, much of this is unpublished, but it appears that there's these PCB52s and especially these metabolites are targeting astrocytes. It appears that the mitochondria is also a major target here. Now, what exactly in the mitochondria, we don't know. And why is this? Is this just is this some interaction of these PCBs, metabolites with, say, individual complexes or with the membranes? We don't know, but we would like to figure that out. And then, but I'd say this work raises a lot of questions too, in terms of, what about microglia? What about other cell types? And so now we're moving on to, mixed cultures of glia. If we irritate the astrocytes, what does that do to the neurons? Is this some kind of a sequential event where the astrocytes are the most sensitive, and then it activates microglia, or is the neurons? We know nothing about the in our experience about the microglia here, but they may have a really important role. All right, I'm going to change gears a little bit for this last bit of the presentation here, and continue on this theme of metabolism, metabolites, and biomarkers, and tell you a little bit different story in terms of again thinking about cell selective toxicity mechanisms, metabolites, and then I do want to draw it back to biomarkers, and so my one of the other projects in the lab has to do with Parkinson's disease, and many of you are familiar with the signs, the symptoms, and there's really there's several needs in terms of the research in this area. One is being able to diagnose it earlier. I mean, if you look on here, symptoms pathogenesis start at some earlier stages, maybe years, decades, but disease may be diagnosed, and you see this classic triad of symptoms, motor symptoms, but non-motor symptoms may be occurring years earlier. However, they are not they're not diagnostic. Loss of sense of smell, GI irregularities are well known. Loss of sense of smell also maps to other neurodegenerative conditions, changes in REM sleep. So there may be some leads here, but really what's needed is something that, biomarker that's reliable, it's quantitative. The other main need with Parkinson's disease is better therapeutics. When you think about Parkinson's disease, L-DOPA is the gold standard drug. It's been the gold standard drug since the 1960s. There's been some attempts in terms of monoclonal antibodies towards alpha-synuclein that have not been effective. The drugs, there's a couple agents that may have a disease-modifying effect, but the therapeutics out there mainly treat symptoms. The dopamine agonists, or like the amantadine, which is maybe effective, and it's antiviral, which is found to be effective in Parkinson's disease, used to control L-DOPA-induced dyskinesia symptoms, and so monoamine oxidase inhibitors, and so the drugs are basically used to treat symptoms. So we need better therapeutics. We also need better ways to diagnose the disease earlier. Here we go. So in terms of Parkinson's disease, the loss of dopamine-producing neurons seems to be the nigrostriatal pathway primarily. You see formation of these Lewy bodies, these protein aggregates. There's also oxidative stress is thought to be a major component, neuroinflammation, these lipid peroxidation products. Dopamine neurons, as many neurologists say, are rare. They're rare. Dopamine itself is a fascinating neurotransmitter. It wasn't until the 1950s that people figured out that it is actually an important neurotransmitter. But for years, it was just thought that, well, it's just an intermediate in the biosynthesis of norepinephrine, epinephrine. Those are the real neurotransmitters. But dopamine is really, really important. We saw the nigrostriatal pathway and other pathways, mesolimbic, mesocortical, transcendibular, very important. So very important neurotransmitter. So in terms of the Parkinson's disease here, in terms of risks and associations, genetics may have a role. Several genes have been identified. I was chatting with someone yesterday, presented at the ADPD conference. This Alzheimer's disease, Parkinson's disease conference that happens every year I've been to it. A lot of the focus has been on genetics, and I think we've learned a lot of really interesting information about the genetics. But one area is still a bit enigmatic, is not a bit, but is enigmatic, is the role of the environment. How do environmental factors serve as risk factors? Which agents here? Look at some of these structures. There's epidemiological evidence that these are risk factors for Parkinson's disease. Epidemiologists say, well, what's the strength of the risk? But these appear to be risk factors. You look at these structures, you see paraquat, highly charged, you see gialodrin, this organochlorine that does interact with GABA receptors. Benamil, interestingly, is a fungicide. It's an aldehyd dehydrogenase inhibitor. There's some recent work in the last 10 years on that, and used in the California Central Valley. And so asking a question, is there a common target for structurally diverse compounds? And so we've asked the question, well, could this be dopamine metabolism and trafficking? And why is that? And just, this is a Goodman and Gilman figure, but look at dopamine, the life cycle of dopamine, its biosynthesis, its packaging in vesicles, it's released into the synapse, reacting with D1, D2-like receptors here, its effector response. And we can see this reuptake here, dopamine transporter or NET, and then metabolism to this acid product, methylation, homovinylic acid, and DOPAC, HPA may show up in the serum here as endpoints. But what's interesting about dopamine is that, it's packaged in these vesicles for reasons for synaptic signaling, but also it's for cytoprotection, because if dopamine is not where if dopamine is located where it shouldn't be, it causes problems. So dopamine, of course, is this catechol group, auto-oxidizes, makes reactive oxygen species, makes these really reactive quinones as well too. And so if you have your dopamine, meaning the cytosol, you're going to see ROS forming, you're going to see thiol modification of proteins, or the dopamine is going to collapse on itself, start forming some pigments, or solution will turn gray, and they'll turn brown. Yeah, it can be a very reactive molecule. And so these vesicles, the pH is a little bit lower, protonated catechols, keeping the molecule happy. And so there's been a fair amount of work with dopamine and the quinone and its toxicity. What's interesting is that I showed you the earlier slide. Dopamine undergoes this reaction via metabolism via monoamine oxidase. Monoamine oxidase catalyzes oxidative deamination. Initially, you form this aldehyde, dihydroxyphenylaldehyde. You form this for things like norepinephrine, epinephrine as well too. This dihydroxyphenylglycolaldehyde. Serotonin undergoes similar pathway, hydroxyindoyl acetaldehyde. You form these aldehydes, which undergo further metabolism, mainly via aldehyde dehydrogenases for dopamine pathway. For epinephrine, norepinephrine, it's actually the reductases predominate. They form these acids and further metabolism. And what's been really fascinating about these this pathways, these aldehydes have been known about for years. Shortly after some initial papers on dopamine is important, they said, hey, it's metabolized in this aldehyde. These things are probably really harmful, but well, we've got the quinone. That's really important too. And so we've spent a fair amount of work thinking about these metabolites, and I'm just going to summarize findings from other labs and from ours, and then jump to this topic of biomarkers. And so, so proper dopamine trafficking metabolism is really important for dopamine neurons. Also, too, what I've completely ignored here, even though I just talked about astrocytes, astrocytes may have a major role as well too. At least, astrocytes take up dopamine, maybe via the DAT or the NAT, to take up dopamine, and they metabolize dopamine. There's the monoamine oxidase, probably in humans more than more B. And so, a lot of the MAO inhibitors are directed against B for various reasons. But yeah, so there's been a lot of animal models throughout the years that have shown that, okay, if you look at dopamine metabolism, you think back to that Goodman and Gilman slide, MAO, VMAT2, aldehyde dehydrogenase. If you start, tampering with these components, you see a common endpoint, progressive loss of dopamine neurons. And so if you overexpress monoamine oxidase, this was Julie Anderson at the Buck Institute, she saw a progressive loss of dopamine neurons in her animals. If you have decreased levels of this VMAT2, similarly you see loss of dopamine neurons over time. Gary Miller at Emory University, and then we've been collaborating actually with Randy Strong, University of Texas San Antonio. Whereas if you decrease aldehyde dehydrogenase activity in the cell, you also see this progressive loss of dopamine neurons over time. And just to note too, what's interesting about these aldehyde dehydrogenases is that there's a lot of them. There's a cytosolic, there's a mitochondrial form. You see a 1 and a 2, but that's only part of the picture. So if you knock out 1 and 2, you've lost maybe 60% of the cellular ALDH activity towards these biogenic aldehydes, or these aldehyde neurotransmitter metabolites. But that seems to be enough to cause this progressive loss. And there's others that publish on this. We publish on this too. I'd be happy to chat with you about this, show you some of our papers. We've shown that this aldehyde of dopamine, it's it's toxic too. It's highly protein reactive. You got a catechol married to an aldehyde. Very, very strange chemistry, biochemistry, highly reactive, physiologic pH. It can make the quinone, but it makes the free radicals, and some of our papers here, some really fascinating biochemistry. But really and so there's this phrase that's been coined, David Goldstein at NIH NINS has referred to this as the catecholaminergic hypothesis of neurodegenerative disease. And so there's thought out here, and there's that perhaps these aldehydes are a component piece of the puzzle for neurodegeneration. And so we've embarked on that pathway too, but also asking a question here, what about a biomarker? And so if you think about, these aldehydes and biomarkers, some of you have done work with 4-hydroxynonenal, malondialdehyde. Anyone has done work with those aldehydes? And anyone here? Maybe not, but there's biomarkers. People have seen these conjugates show up in serum and urine. You said, well, maybe we can use these aldehydes as biomarkers. And actually, what we've shown too, just to ask this question of how are these aldehydes generated at aberrant levels? Why is that a problem? And what we've shown over the years here is that products of oxidative stress, if you oxidative stress, lipid peroxidation products, they fundamentally they shut down aldehyde metabolism in the cell, biogenic aldehyde metabolism. The aldehyde dehydrogenases, especially things like malondialdehyde, will inhibit the aldehyde reductases. At lower concentrations, these products of oxidative stress, lipid peroxidation products, it appears to be reversible. Higher concentrations, it seems to be, we see some irreversible binding, but oxidative stress will shut down this metabolism. Carbonyl metabolism seems to be sensitive. Environmental agents, dieldrin, organochlorines, chlorpyrifos. We also see changes in terms of the metabolism here. And so one question I just want to plant seed and plant now is in terms of these aldehydes. Okay, so it's is this somehow piece of the puzzle for disease? But in terms of biomarker, that's a great thought. But how are you going to measure it? And it's uniquely reactive. If you read some of these papers, you'll see, okay, wow, this thing is uniquely reactive, it's highly reactive, it's uniquely reactive. So how can you measure it? And so we can measure it in tissues. We've done it in numerous tissues, in zebrafish and mice, mouse brain, rat brain, C. elegans. We measure these aldehydes. But electrochemical detection, I'm never quite certain what is the bound versus the free. What I'm seeing free here, is this really representative of how much is actually produced? And we've tried to make antibodies. We had an antibody that worked okay for dot blots, but it was useless for Westerns. We're going to try again. So we're limited in some of our technology. If anyone has any ideas on that, a detect, capture, be much appreciated. Or how to make a good antibody, that would be fantastic. That's one thing that's been hugely limiting to us. Many of the other biogenic aldehydes, like lipid peroxidation products, are fantastic antibodies. So, all right, so biomarker, how in the world are we going to measure it? So, all right, so in terms of the production of this aldehyde at aberrant levels, just a few quick slides here. We've seen things like chlorpyrifos and its auxin. If you treat dopaminergic cells, you see this dopac, which is the acid product that goes down, and you see elevated levels of the chlorpyrifos. You see corresponding increase in terms of reactive oxygen species formed as well. I'll go to the next slide here. And it's interesting, we do see two of these cells that here's this chlorpyrifos auxin and these dopaminergic cells. These are the N27 cells. We see that there seems to be some dopamine dependence in terms of its toxicity. I just to note that these N27 cells are dopaminergic. One thing that is problematic about them is they don't express much dopamine transporter. We actually have a variant of it now that stably expresses a human DAT, which has been a really wonderful model, but I won't talk about that today. So all right, so we see production of these this dopal here, these aldehydes, in particular this dihydroxyphenylacetaldehyde or dopal, in response to environmental agents, dieldrin. You can get some of our work on that. And so that's a question. Well, can we? Is there any biomarkers that we can use? And so we got kind of a lucky break when some years ago I was collaborating with a lab just down the hallway. Was interested in on cardiovascular health and interested in norepinephrine, epinephrine, actually the aldehydes of those neurotransmitters and cardiovascular health. And so we were chatting about, okay, well, is there some scavengers? There's some scavengers. There's been a lot of interest in this dipeptide carnosine as being cardioprotective. And this dipeptide here of beta-alanine, histidine, and there's been myriads of studies on it, and protective antioxidant, chelating agent, kind of a cure-all panacea. But what we found that was really interesting was that this dipeptide is actually a really good scavenger of this dopal. It reacts quite well with this dopal, forming a stable adduct. And what's interesting is if you look at this graph here, so 4-hydroxynonenal is a lipid peroxidation product, an alpha beta and saturated carbonyl, and we've got our dopal. And if you incubate these two biogenic aldehydes with carnosine, you see loss or reaction of dopal with this carnosine and just a little bit of reactivity with this 4-hydroxynonenal. Not that impressive, but you look in the literature, you'll see that there's reaction and these two bind. So it seems like this carnosine is a better scavenger of this dopal, and there's this 4-hydroxynonenal. But if we shift gears and look at the wonderful tripeptide glutathione, if you look at this, what you see is that glutathione is highly reactive towards 4-hydroxynonenal. This is without any GST. This is just straight up glutathione. It's, Michael reaction, very, very reactive. Within minutes, we see consumption. But look at this dopal. There's no reaction of dopal with glutathione. These two don't react. There's an interesting side story I don't have time to tell you about, but thiols seem to stabilize this dopal as an antioxidant. But what we also found was that if you take L-cysteine and react and add it to this dopal, or carnosine, you see this very rapid reaction of dopal with L-cysteine. And L-cysteine is actually a classic aldehyde trapping reagent. You got the amine, you got the thiol, this chomps down on the aldehyde here. We saw this reaction occurring very, very rapidly. And move forward here. And so a student, Rachel, found with our QTOF, I have a love-hate relationship with. It's got great mass accuracy. When it's working, it works great. When it's not, it's just a pain. What she saw was that, with dopaminergic cells, you give them some N-acetylcysteine, you give them some dopamine, they start producing this conjugate, this L-cysteine dopal conjugate. So what's happening is the N-acetylcysteine is being converted to L-cysteine, the cells, the dopamine is being converted to the aldehyde, and you're forming this conjugate. This conjugate form seems to be exported to the extracellular compartment. We see very, very little in the intracellular, whatever that's worth. And so what she's kind of producing is kind of like this homegrown conjugate here, where the dopamine has to get into the cells. It's converted to the aldehyde. The N-acetylcysteine gets in, converted to the L-cis. We form the conjugate. It's exported. At this point, we can't completely rule out extracellular conjugation, but at least these processes are intracellular, and we can see time-dependent formation with this L-cis conjugate here. What was interesting was she found that if you took cells, dopaminergic cells, and you load them up with a little bit of dopamine and you stress them, she saw formation of this conjugate. And actually, what she saw was that, and with a QTOF, we're able to get this wonderful lock on this conjugate, and fragmentation can confirm it. And what we saw was that, if you take the cells, give them a little bit of dopamine, 1 micromolar dopamine, and then you stress them, and with a couple, things like, biphenyl, or we saw rotenone here, after 3 hours, you start seeing significant formation of this conjugate, which appears to be stable. Now, some questions remain, and we're actually in the process of trying to move this to in vivo model with aldehyde dehydrogenase knockout mice, try to push the system to find this conjugate. One thing that we run into is that this conjugate eventually undergoes further processing. And so at least one of the initial products here is this methyl, probably catecholaminergic transferase. You see this methylation of this conjugate. And so we've looked at some other human serum, and we've tried pushing this reaction with human liver microsomes, and we think that there's further metabolism going on. We don't quite know at this point. So yeah, so just in terms of what's going on here, in terms of overview, again, we've got these two themes here: cell-selective toxicity, PCBs, organochlorines, potentially astrocytes, mitochondria as being a target. and then shifted gears to tell you about this neurotoxic metabolite and biomarker. And again, thinking about mechanisms of toxicity here, toxicants interfering with endogenous processes, again, yielding toxicity, disease, and then biomarkers. And then, here we go. Oh yeah, and this ongoing collaboration with Randy Strong, looking at Doorn and ALDH1/2 knockout mice, seeing if that potentiates the toxicity. Interestingly, we're seeing, it looks like the glia may be targeted with the Doorn, more to come. And just to think, right now I've got some wonderful students, Nia, Anna, Michael, Rachel Crawford just defended, took a postdoc at Johns Hopkins, have had wonderful collaborators, on campus, from the Superfund, Dr. Hans Lemler, Laura Dean, Andrea Martinez, other collaborators too that help us with cell models. At heart, I like thinking about mechanisms. I like starting with simpler models and then building complexity. And so for me, it's been really an adventure to learn more about mitochondria, astrocytes, and eventually, how do we put this all together? How are cells interacting with each other? How are they talking to each other to, yield toxicity as well as disease? Also, just a picture of our brand new College of Pharmacy at the bottom right-hand corner. Right now, the live stream picture would have 18 inches of snow and gray skies, but this is from a better day, probably I think in the summer. Actually, I've never seen the Iowa sky that blue. So, all right. So with that, thank you for listening to me. I've taken a full hour. Be happy to have any questions.